students, we are doing the 11th chapter, that is transport in plants. And students, today we will conclude this chapter by doing the last topic, that is uptake and transport of minerals and phloem transport. Students, we all know that water is absorbed by two ways. One is passive absorption and another is active absorption. In passive absorption, the water molecules, they move along the concentration gradient and no energy is involved. Whereas in case of active absorption, the water molecules, they move against the concentration gradient and energy is involved. Students, mineral ions, they are also absorbed by two ways like water. One is passive absorption and another is active absorption. But unlike water, all the mineral ions, they are not absorbed passively. Now, why all the mineral ions, they are not absorbed passively? First reason is this, that the mineral ions, they are charged particles and they cannot cross the cell membrane. Second reason is this, that the mineral ions which are present in the soil solution, they are in lower concentration as compared to the mineral ions which are present within the plants. So these are the reasons that mostly the mineral ions, they are not absorbed passively. So how they are absorbed? They are absorbed actively. It means energy is involved in the process of absorption of mineral ions. And mineral ions, after the absorption, they are moving by two pathways. One is the apoplast pathway and second is the symplast pathway. Students, we all know that in apoplast pathway, the molecules, they move from one cell to another cell through the cell wall, whereas in case of symplast pathway, the molecules, they move from one cell to another cell from plasmodesmata, that is cell to cell connections. In plasmodesmata, the protoplasm of the one cell, that is continuous with the another cell. So in symplast pathway, the molecules, they are moving from protoplasm of one cell to the protoplasm of the another cell. So mostly the mineral ions, they are absorbed from the soil solution actively. Now students, our next topic is translocation or phloem transport. Students, we all know that food is manufactured by the green leaves by the process of photosynthesis. But the food which is manufactured by the green leaves, that is actually needed by all parts of the plant body, by the non-green cells also, for their growth and metabolism. So this movement of food particles from the leaves to all the parts of the plant body, this is known as what? Translocation. And what is the pathway of translocation? The pathway of the translocation is the phloem, and the movement of food, that is bidirectional as compared to the movement of water in the xylem, which is unidirectional. So students, today we will be discussing on these topics in detail.
Dear students, the absorption of minerals takes place by both passive and active mode, but mostly the minerals, they are absorbed by active mode. It means energy is involved in the absorption of mineral ions. Why? Because the concentration of mineral ions, that is found to be higher within the plant body. So energy is involved in the absorption of mineral ions from the soil solution to the plant body. And this is known as what? Active absorption. And once the minerals, they enter within the epiblema, then with apoplast or symplast pathway, they are entering into the phloem part. Now, what is translocation, students? Let us study it. We all know that organic materials, they are produced by the plant body as a result of photosynthesis. And this organic food, which is synthesized by the plant body or by the green leaves, they are to be transported to other regions of the plant where it will be used for the growth or it will be stored. So this process of movement of organic materials from the leaves to all the part of the plant body for growth and for storage, this is known as translocation. And the pathway of translocation is the phloem. So phloem is responsible for translocation. So Translocation is the long distance transport of food materials within a plant body. We all know that plants, they are manufacturing food and they are synthesizing food in the form of glucose. This glucose, which is produced in the leaves, that is converted to sugars, mainly sucrose. And this sucrose is then translocated to different parts of the body. So the sucrose which is synthesized within the leaves, that is translocated to storage organs, for example to fruit where it will be stored mainly as sugar, or it is translocated to the upper meristematic part, to the growing regions where it will be used for the process of growth, or it is also translocated to the lower part of the plant body, that is to the roots, where again the food that is stored. So these are the various regions where the food which is synthesized in the leaves, that is translocated. And the pathway of the translocation, it is the phloem. So we can see here that the direction of translocation, that is, it is in both direction, both in upward as well as in the downward direction. Whereas the movement of water in case of xylem, that is only in one direction, upward direction. But the movement of food materials, that is taking place in both the direction, in upward as well as in downward direction. So there is bidirectional translocation within the phloem. Now what is the evidences which prove that it is the phloem which is the pathway for the translocation of organic solutes? Now when phloem is cut, the exuded sap, it is rich in organic materials such as carbohydrates. So when the phloem is cut, then a sap, it starts coming out from it, and this sap, it contains carbohydrates. What does it mean? It means that carbohydrates, they are moving through the phloem. The sugar content of the phloem, it varies in relation to the environmental conditions. And one of the most important experimental evidence which proves that it is the phloem through which the translocation that is taking place, this is ringing or girdling experiment. What we do in ringing or girdling experiment? In ringing or girdling experiment, a ring of bark that is removed, and this bark, it contains the phloem from the stem. So what will happen? The food, it will start accumulating above the ring, and it will cause cessation of the downward movement of the substances. So let us study this experiment with the help of some pictures. So to show the site of sugar movement in a plant, we will perform ringing or girdling experiment. And what we do in ringing or girdling experiment, a complete ring of bark which contains the phloem tissue, that is removed from the woody stem. So we have removed the bark, and this bark, it contains the phloem. It means we are interrupting the passage of the movement of food materials. If it is phloem through which the movement of food materials, it is taking place, so we have interrupted its path. Now the upper part, it is still connected with the lower part. We have only removed the bark portion, but the upper part, that is still connected with the lower part, with the xylem or with the pith tissue. It means the upper part, that is getting water or minerals from the lower part. So what will happen after some time when a ring of bark that is removed? Then what will happen? The removal of the phloem, it will prevent the transport of food materials to the lower part of the plant body. So what will happen? The upper part of the plant body, that is synthesizing food materials. Now when this food, it will move downwards. Now there is no phloem in the lowermost part. So the food materials kya honge students? They will start accumulating above the ring, and the upper part of this ring, it will get swollen. because the food materials, it will get collected here, and after some times even the adventitious roots, they start appearing from this part. So क्या होगा students, we have interrupted the path of the movement of the food materials. So रिंग के ऊपर जो पार्ट है वहाँ पे फूड जो है वो एक्यूमुलेट हो जाएगा, वो पार्ट जो है वो स्वोलन हो जाएगा, और वहाँ से एडवेंटिशियस रूट्स जो हैं इट विल स्टार्ट अपियरिंग, and the tissues below the ring, they will begin to die. Why they will begin to die? Because they are getting no food materials from the upper part, because we have removed the phloem part through which the translocation of food materials it is taking place. So this experiment, that is ringing or girdling experiment, it confirms that it is the phloem through which the translocation that is taking place. Students, even you can perform this experiment at your home to prove that it is the phloem through which translocation it takes place. So what is the conclusion of this experiment? It is the conclusion that it is the phloem tissue which is the site for the sugar transport. So this experiment, it confirms that it is the phloem which is the pathway for the movement of organic solutes. Now students, another experiment is this, कि if radioactive carbon dioxide that is given to the plants, then carbohydrates which will be formed within the plant body, that will also become radioactive. And these radioactive carbohydrates, they are moving within the phloem. If they are moving through the phloem, then radioactivity, it will be detected in the phloem part. So if radioactivity it is detected within the phloem part, it confirms that it is the phloem through which the translocation that is taking place. So students, इस experiment में क्या करते हैं, radioactive carbon dioxide जो है that is given to the plants. So plants, they will use this radioactive carbon dioxide to manufacture the food. And what happens by the autoradiography technique, we find the accumulation of radioactivity. So it is found by autoradiography technique that the whole of the radioactivity that is accumulated in the phloem part. It means it is the phloem through which the translocation that takes place. Now students, next है using aphids. So using aphids also we can, it is shown that it is the phloem, because the aphids, they penetrates the stem into the phloem using its mouth parts called stylets. So stylets जो है, it can pierce the stem up to the phloem and it sucks the plant sap. So a feeding aphid के जो stylet है, that is cut, and from that cut stylet, the phloem sap, it starts flowing out. And when it is analyzed, it is found that in this phloem sap, sugars and other organic substances, they are present. So this also proves that it is the phloem through which the translocation that is taking place. So these are the various experiments which were performed to prove that it is the phloem through which translocation happens. Now what is the mechanism of translocation? How translocation it takes place? We all know that food that is manufactured in the green leaves. So there is high level of sugar at the source, which will lower the water potential and it will cause movement of water or flow of water into the tube. So leaves जो हैं, they are manufacturing their food. So because of the accumulation of the solute particles, there is lowering of water potential. So water, it will enter into the phloem part from the xylem. And what is happening at the sink region? Sink regions वो parts हैं जहाँ पे ये glucose जो है वो either stored हो रहा है और it is used for the growth purpose. So यहाँ से glucose की जो है वो removal हो रही है. So water potential जो है it increases. So it will cause the flow of water out of the tube. So this is the mechanism of the translocation. भी source जो region है वहाँ पे water potential की value low है और जो sink region है वहाँ पे water potential की value जो है वो high है, and this is causing the movement of the water molecules along with the solute particles. So very important students, the mechanism of translocation that was explained by various experiments or by various theories, but the most widely accepted is the Munch mass flow hypothesis, which was given by Ernest Munch in the year 1930. So he proposed a hypothesis which is known as mass flow hypothesis or pressure flow hypothesis to explain the mechanism of the translocation of organic solutes. So this is very important hypothesis. So Munch, with the help of an experiment, he proved the movement of the solute particles through the phloem and how they are moved from the source region to the sink region. So what Munch performed, he took two osmometers. So Munch ने दो osmometers लिए, osmometer A and osmometer B. In osmometer A, there is high concentration of the sugar, whereas in case of osmometer B, there is low concentration of the sugar. और दोनों osmometers को connect कर दिया गया with the tube C. Now students, in case of A osmometer, there is high concentration of solute. So what will happen? Water, it will enter within this osmometer by the process of endo-osmosis. Because there is increase in the concentration, in osmotic concentration within the A osmometer, so naturally water जो है वो enter करेगा by the process of osmosis, and there will be increase in the turgor pressure within the osmometer A. Now in case of osmometer B, there is low concentration of solute. So here also the water, it will enter, but the turgor pressure will be low as compared to the osmometer A. So in case of osmometer A, there is high turgor pressure, whereas in case of osmometer B, there is low turgor pressure. So there is a difference in the turgor pressure along the two osmometers. And this difference in the value of turgor pressure along the two osmometer, this is known as turgor pressure gradient. And this turgor pressure gradient will cause the mass flow. Mass flow means movement of water along with the solutes from the osmometer A to the osmometer B through the tube C. क्लियर स्टूडेंट्स? सो इस तरीके से मंच ने जो है वो ट्रांसलोकेशन मैकेनिज्म को एक्सप्लेन किया था. सो वी विल स्टडी इट फर्दर विद मोर एक्सप्लेनेशन. सो मंच ही टुक टू osmometers, osmometer A and osmometer B. In osmometer A, there is high concentration of sugar, whereas in case of osmometer B, there is low concentration of sugar. So the water, it will enter into the osmometer A by the process of osmosis, and the turgor pressure, it will get increased. This increase in turgor pressure within the osmometer A, it will cause mass flow of sugar solution. Mass flow का मतलब क्या है students? Then water will move, and along with water, the sugar molecules, they will also start moving. So this increase in turgor pressure will cause the mass flow of sugar solution to osmometer B through the tube C under the influence of a gradient which is known as turgor pressure gradient. Now, the water from B that is forced out because of the pressure created, because एक pressure create हो गया है, turgor pressure gradient जो है वो create हुआ है between the two osmometers. So water from osmometer B that is forced out because of the pressure created, and the movement of solute will continue. Now there will be movement of solute and it will continue till the solution in both the osmometers, they attain the same concentration of solute. मतलब तब तक मास फ्लो होता रहेगा वाटर का along with the solute particles until the concentration of the sugar solution it becomes equal in both the osmometers. और इस process को हम prolong भी कर सकते हैं if a continuous supply of sugar solution in osmometer A is maintained. अगर A जो है osmometer उसमें हम sugar की concentration जो है वो increase कर रहे हैं, so this process it will keep on moving. सो मंच ने स्टूडेंट्स ये दोनों ऑसोमीटर्स को कंपेयर किया है. मंच हैज कंपेयर्ड द ऑसोमीटर ए to the supply end, that is to the leaves which are manufacturing food or solute particles, and he has compared the osmometer B to the consumption end, that is to the root part or to the storage organs. और ट्यूब सी की कंपैरिजन किसके साथ हुई है स्टूडेंट्स? फ्लोएम के साथ इसको कंपेयर किया गया है. सो वी ऑल नो दैट लीव्स दे आर मैन्युफैक्चरिंग फूड. So here the osmotic concentration is high. Because of high osmotic concentration, it will absorb water from the neighboring cells. So there will be increase in the turgor pressure. Whereas in case of roots or storage organs, here the food that is used, so there is low concentration of organic solutes. So because of the low concentration of the organic solutes, there is less turgor pressure. So a turgor pressure gradient that is created between the supply or between the source and the sink end, and this turgor pressure gradient will cause the mass flow of water along with the solute particles through the phloem region. So students, मंच ने ये जो एक्सपेरिमेंट परफॉर्म किया, अल्टीमेटली इस एक्सपेरिमेंट के थ्रू उसने ये प्रूव किया दैट हाउ ट्रांसलोकेशन दैट इज टेकिंग प्लेस फ्रॉम द सोर्स एंड, दैट इज फ्रॉम द लीव्स to the sink end, and the sink end, they are the parts like they can be roots or they can be storage organs or they can be meristems where growth that is taking place. So what is happening? Photosynthesis जो है that is taking place in the mesophyll cells, which will result in the formation of sugars. So what will happen? There is increase in the osmotic concentration. So because of the increase in the osmotic concentration, there will be increased osmotic potential. And if there is increase in the osmotic concentration, so what will happen students? Ki within the leaves, the food that is manufactured. Now this food, it will be transported to the phloem part. और यहाँ पे ये जो ट्रांसपोर्ट हो रहा है फूड मटेरियल्स का, इट इज़ एक्टिवली. सो फूड मटेरियल्स जो है वो एक्टिवली ट्रांसपोर्ट हो रहे हैं फ्रॉम द मेसोफिल सेल्स टू द फ्लोएम रीजन. सो फ्लोएम रीजन में हुआ क्या? फ्लोएम में जो है सीव ट्यूब्स जो हैं वो चैनल्स का काम करती हैं. और देखो स्टूडेंट्स, ये जो सीव ट्यूब्स हैं दे आर प्लेस्ड वन अबव द अदर, एंड इन सीव ट्यूब्स के जो एंड्स हैं दे आर परफोरेटेड, दे आर नोन एज सीव प्लेट्स. और सीव प्लेट्स के थ्रू स्टूडेंट्स एक सीव ट्यूब का साइटोप्लाज्म जो है दैट इज कंटिन्यूस विद द साइटोप्लाज्म ऑफ द अदर सीव ट्यूब्स. सो दीज सीव ट्यूब्स दे आर एक्टिंग एज लॉन्ग चैनल्स थ्रू विच द फूड मटेरियल्स दैट इज ट्रांसलोकेटेड. सो फूड जो है वो मैन्युफैक्चर्ड हुआ विद इन द मेसोफिल सेल्स. ये फूड जो है फिर एक्टिवली ट्रांसपोर्ट हो रहा है इनटू द फ्लोएम रीजन. Now there is increase in the osmotic concentration within the phloem part, so it will cause absorption of water from the xylem. So there will be absorption of water from the xylem, and sugar, it will start dissolving in it, so there will be increase in the turgor pressure. At the same time students, in the cells of the roots or the storage organs, the food materials, they are consumed or they are converted into the insoluble starch. So what is happening there? There is increase in the concentration, there is decrease in osmotic concentration, and the turgor pressure, it will be low at the sink area. So in this way, a turgor pressure gradient that is created or that is formed between the supply end and the consumption end, and as a result of this, water with the dissolved food materials, it will move passively. Okay, now there will be passive movement of water along with the solutes from the supply end to the consumption end, and in this way students, translocation that is taking place. So students, how in the end I will tell you that how the translocation that is ultimately taking place. Food that is manufactured in the source area, that is in the green leaves, And this food, which is manufactured within the green leaves or in the mesophyll cells, that is transported to the phloem region by active mode. Now there will be increase in the osmotic concentration within the phloem part. So this will cause the movement of water from the xylem into the phloem. So what will happen? There will be increase in the Tugger pressure value. नाउ स्टूडेंट सिंक एरियाज जो हैं, फॉर एग्जांपल जो रूट एरियाज हैं या स्टोरेज ऑर्गन्स हैं, वहाँ पे फूड मटेरियल्स जो हैं वो यूज़ हो रहे हैं या वो स्टोर्ड हो रहे हैं. एंड वी ऑल नो दैट ऑस्मोटिक जो कंसंट्रेशन है या ऑस्मोटिक प्रेशर जो है, दैट इज़ अ कॉलिगेटिव प्रॉपर्टी. वो सल्यूट पार्टिकल्स के नंबर के ऊपर डिपेंड कर रही है, नॉट अपॉन देयर नेचर. सो जो सोर्स एरिया है वहाँ पे सल्यूट की कंसंट्रेशन ज़्यादा थी, तो वहाँ पे ऑस्मोटिक प्रेशर और टगर प्रेशर क्या था? हाई था. सिंक एरियाज में क्योंकि फूड मटेरियल यूज़ हो रहा है, तो यहाँ पे ऑस्मोटिक प्रेशर की वैल्यू लो है और टगर प्रेशर की वैल्यू क्या है? लो है. So there is a difference in the value of the Tugger pressure along the two sides, that is between the source area and in the sink area. So ये जो difference है in the Tugger pressure value, this will cause the mass flow of water along with the solute particles from the source end to the sink end, and in this way students, translocation of food materials that is taking place. So I hope students, with the help of Munch flow hypothesis, we have clearly understood how the translocation or what is the mechanism of translocation. So students, this was all about today's topic. So we have studied in detail about the various experimental evidences by which it was proved that it is the phloem through which the translocation it is taking place, and we have clearly understood what is Munch mass flow hypothesis, which was which was done by Munch to explain the mechanism of translocation. So students, this was all about today's topic. Thank you all of you.